Okay, we're gonna get started. First off, welcome to everyone who is here. We are so glad to have you all, and hope that you enjoy the presentation that is happening tonight. We have a great presentation for you all from our student researchers as well as our awesome professor. So first, I want to introduce them to you. I'll introduce the lab. So first, you need to know a little bit about the professor who was leading the charge, Dr. Philips Akinwole. He earned his master's in environmental science from IHE, the Institute for Water Education, which is in the Netherlands. And he spent several snowy days scouting streams in the German Black Forest for his thesis. He then earned a doctorate in microbial ecology from the University of Alabama, Tuscaloosa, where his research examined microorganisms involved in dissolved organic carbon utilization in the Catskill Mountains. Currently at DePauw, he teaches microbiology, molecules, genes, and cells, and the discovery processes in science and math. He is a microbial ecologist and asks questions on how spatio-temporal variations in environmental gradients drive ecological patterns and processes, especially in urban populations. There is an emphasis on anthropogenic impacts, which are man-made pollution, and how those influence species distribution and biodiversity patterns. Apart from teaching and research, he enjoys trying new dishes, traveling, and visiting new places. Now, for the students, Amerti Guta is a junior cellular and molecular biology and biochemistry double major. She is an international student from Ethiopia and joined Dr. Akinwole's lab during her first year at DePauw. As a student researcher, she is interested in ecological microbiology and the connection between ecology and infectious diseases. She's also part of Dr. Gernan's lab that looks into in silico prediction of benign and pathogenic mutations. On campus, she serves as a resident assistant, a STEM guide, and a lab assistant. She's also a civic fellow, and she's passionate about research and mentoring. With her interests in microbiology and structural biology, she ultimately hopes to combine a career in infectious diseases. Upon her completion of study at DePauw, she plans to pursue her PhD and MD in infectious disease. Sophia Atkinson is a senior biology major and German minor from Connorsville, Indiana. She's a Lilly Scholar who was awarded the Truman G. Juncker Award as an outstanding biology major and was also awarded the G. Hans Gruinger Prize for high achieving students in German studies. With an interest in conservation and restoration studies, she has worked in the local Greencastle Big Walnut Creek and the DePauw University Nature Park, studying how anthropogenic activities have affected local organisms, including microbes and plant life. She continues to study how microbes are affecting the restoration of the abandoned quarry through biological crusts. She pairs her research with sustainable action as a student director of the Sustainability Leadership Program, a student-led organization interested in meaningful, sustainable changes to DePauw's campus and the greater Greencastle community. And then finally, last but not least, Maddie Draper is a senior DePauw student from Chicago, Illinois. She is majoring in cellular and molecular biology and is planning on getting her master's in pathology assistance. During her time at DePauw, Maddie has been a part of the cross-country team as well as the track and field team. She is a science research fellow and a student intern for the Alumni Development Department, so she has a soft spot in all of our hearts here in the alumni office. Maddie has been doing research with Professor Akinwole since her sophomore year. Through her research, she has had abstracts accepted at 2 conferences. She has co-authored a research paper and is currently working on having her second paper published. So that, my friends, is our research team. And I will now hand it over to Dr. Akinwole to explain the research and kick it off with this awesome presentation.
Good evening, everyone, and I want to thank the organizer of this program for giving my lab the opportunity to talk with our alumni. And we also want to thank our alumni for all the support and the funding that we already got from them. And so thank a lot for coming around to listen to us. I joined DePauw in the fall of 2018, and I found this place to be very conducive for my work and also engaging students. Right from the fall semester, I got funding to start my works, and I was able to take students to neighboring state to collect white clover plants. So I wasn't sure how the students here are they going to kind of be able to work with me, and how they're going to, the kind of experiment that I'm doing. But I found out that they were so excited, very happy, and very eager, to get out of the lab, the classroom, and to walk outside. And so we were collecting white clovers all over, the Midwest. And so the power was kind of that time, I found it very encouraging to me to start working right from my fall semester. So as a microbiologist, so my interest is to look at the connection between anthropogenic impacts and the ecosystem function that microbes perform. And so then I applied for another grant, and I got another grant the following year. And that was when I was able to get my student, Amerti, Sophia, and Maddie to work on the Big Walnut Creek. And then it was difficult to find any information on the Big Walnut Creek in the literature. So we have to do all that information, and then so I have to look for a local person to kind of help me to kind of do the initial survey of the Big Walnut Creek. And then that's how we started, and then we were able to map out several spots for our study. And then we want to see the impact of the wastewater effluent on these microbes in the Big Walnut Creek, because we know that the microbes are very important in our life. So a lot of people think about diseases when you hear about bacteria, but then without the bacteria, we will not all be alive right now. They the one that provide the oxygen that we breathe. They are the one that help us with leaf decomposition. They're the one that recycle all the elements. Talk about nitrogen, carbon, oxygen, sulfur, phosphorus, all those elements. So these microbes recycle them for us so that we can enjoy the nature. But then because of several activities of pollution and chemical that we input back into this ecosystem, we are impacting the microbial life. Because the microbes are very sensitive to pollution, so we can look at the changes in their community or in the structure or in the diversity, and then from there we can see what is going on, how we are affecting them, and how can we correct our mistakes. So that's why we're kind of interested in the microbial community and what we are doing to them. And so my job is to monitor who is there in the Big Walnut Creek. How do they change? What change their community? What change their composition? What are we doing to them? Are this community the same in summer, in spring, in winter? And what factors can influence them? So we study stuff like that. And then we try to correlate that to public health. and how that can affect the ecosystem function that's so important to us. And then to do that, we have a lot of techniques that we have to do in the lab. And as a new faculty, I was worried, oh, will these students be able to do all these complex techniques, biomass, DNA extraction? I don't want them to screw up my lab. So but then I have awesome students. I think I walked them through the first time and then they just got it. And okay, Sophia, go ahead and do all the biomass. You're awesome. Go ahead and do the DNA. And so Maddie and the Amerti and so students here, they're very eager, they're very happy to work, and they're ready to learn new techniques and they're just kind of awesome. I've enjoyed working with them. I've taken some of them to conferences. So the last one was in 2019, and we won an award. We couldn't go 2021 because of the COVID. We're hoping to go this year, all things be equal. So we're working towards that. So DePauw is a good place, providing opportunity for students to do research work and also providing good education for them. And also, I've been supported to engage students and kind of let them be well prepared for graduate school or for job. So this work, as they're gonna be talking to you tonight, is about how the effluent from the wastewater facility can impact the environment and can also, affect the diversity that we have in the microorganisms that are so important to our life. So we kind of leave the rest to them, and they will tell you all the stories. Thank you so much.
Thank you, Professor. And hi, thank you everyone for coming to the webinar this evening. We will be presenting the paper we published in the summer of 2021, the focus being on the impacts of wastewater treatment plant runoff on microbes in the Big Walnut Creek. Could you go to the second slide, please? Thank you. So I first want to really dive into the importance of studying our waterways. Waterways are important pieces of the environment, being a home to a plethora of different species. You have, of course, your larger organisms like fish, your frogs, turtles, maybe the occasional bird that will stop by, but there's an entire world if you look just a little further into who is there. And that is when you find algae and bacteria and planktonic crustaceans like Daphnia, which is the little guy in the picture in the middle there on the slide. And these species make up a large biodiversity that brings important benefits to the rest of the environment, and many have different services that they can provide. And these services look like the breakdown of leaf litter that accumulates throughout seasons, adding nutrients to the environment, and some microbes may even be involved in carbon sequestration. And so these services are really needed for the overall ecosystem function and sustainability. Next slide, please. So when we talk about wastewater effluent in the Big Walnut Creek, we are looking at the water that has been treated in wastewater treatment plants and then released into the Big Walnut Creek. So sewage and industrial waste comes into the treatment center and goes through a screening process, then onto a primary, secondary, and tertiary treatment stage to then be released into an aquatic environment, which in this case is the Big Walnut Creek. And wastewater treatment facilities also either chlorinate or use UV to disinfect the wastewater. And the Greencastle treatment facility uses UV to disinfect, and it is a lot more environmentally friendly. Next slide, please. So why study the impacts of wastewater effluent? The unfortunate truth to wastewater treatment facilities is that they do not capture absolutely everything when treating wastewater. The effluent released into aquatic environments may still contain heavy metals from industrial waste, pharmaceutical waste from the general population, because any medication you take, your body does not absorb 100% of the ingredients. And then there are also other pathogens and nutrients that may disrupt microbial communities when released back into waterways. And wastewater treatment plants were not designed to capture all of the substances, so they slip through, and this is why we are interested in studying the impacts that they have on the biodiversity and community composition of microbial communities. Next slide, please. So our main research question was, how does the wastewater effluent affect microbes throughout multiple seasons? While doing this research, we were very interested in looking at the total amount of microbes in the areas we sampled through biomass, taking biomass, what structure of these microbe communities look like, and then what kind of carbon sources they used as food. Next slide, please.
Again, thank you very much for attending our presentation, and it's definitely a pleasure to present our work tonight. So with me, we'll go over the sample collection, the methods we used, and also some part of the result. So for our study, we took our samples from 5 locations along 5,000 meters of the Big Walnut Creek. We had 2 upstream locations and then 2 downstream locations. So as you can see here on the map, we have the Dunbar Covered Bridge, which is DCB, serving as our control because it's the first upstream location. Then we have WWD, where the effluent meets the river. And downstream, we have NP, which passed through the nature park, and OCB, which is referred to as the Ocala Covered Bridge. We collected our samples in 2019 and 2020 in 3 different months, which are September, October, and February. Next slide, please. Now, talking a little bit about the experimental procedures we did. The first experimental procedure we did was measuring physiochemical parameters, such as temperature, pH, percent water content, and carbon nitrogen ratio. So determining those physical and chemical factors will enable us to reason out the experimental results we got through our experiment. At every sample site, as you can see here, Sophia and Maddie doing, we used a probe to take a measurement of water pH and temperature. And in order to determine nutrient availability, which also was a huge factor in our research, we sent our samples, dried samples, to a company, which helped us measure the carbon nitrogen ratio. So the level of nitrogen is a major determinant in the amount of nutrient availability. Considering this, if we have a low carbon nitrogen ratio, then we predict that we have a high nutrient availability in that region. Next slide, please. So as we all have our own favorite food, so do bacteria. When we use this concept to categorize a community of bacteria, we refer to it as community-labeled physiological profiling. So as you can see here in the slide, we have a plate with 31 different substrates, or technically food for the bacteria. When the bacteria utilizes the carbon source and begins respiring, the color would turn into purple, as you can see here in some of the wells. However, if the bacteria is not using that carbon source, we'll have colorless wells, as you can see some of the wells in the picture illustrated here. However, this is only a qualitative data, and it didn't provide us the extent of information we're seeking. Therefore, we took the plate and then placed in a reader referred to as the spectrophotometer, and then took the intensity of the coloration. And maybe while describe this, you might be wondering how we were able to employ this into our research. In short, what we did was we took our samples, diluted it, and then added it in this plate, which has 96 wells. And we took the measurements every 24 hours over 10 days. One of the very great things about using the EcoPlate is that it has replicates. We have 31 substrates, so it has 3 different columns of the same substrate repeating. So we were able to get a good amount of data by just doing few plates, which enabled us to have a good representative sample of our sample site. Next slide, please. So the other experimental procedure that was fundamental in our research was the phospholipid phosphate analysis. So in order to study the change in the number of bacteria over the sample sites or locations, we measured the biomass of the bacterial community, which is simply the complete mass of the bacteria. So let's say if we have 5 people in a certain area and then 1 person in a different area, we'll have a higher mass for the 5 people compared to the 1%. Of course, the concept might not be as simple as this, but translating it to our research, biologically, we refer to it as phospholipid phosphate analysis. We employ this method in order to extract the fatty acid in the cell membrane of the bacteria. So looking at this picture, the bacteria has a capsule, then a cell wall, and a cell membrane. And this cell membrane is essentially made out of fatty acid. So extracting this fatty acid and quantifying it will enable us to predict the number of bacteria or the quantity of bacteria in that region. And something really important about this procedure, and that also made us use this procedure, is that it only measures the biomass for viable and living bacteria, which makes it closely accurate and very good representation of the sample site we're looking at. Next slide, please. So starting with the results section now, here we will look at the microbial biomass calculated over 5 sample sites and 3 different ones. We saw a significant seasonal difference as illustrated here in the graph. So looking at the graph right here, the low biomass in February, which is winter, was about 2 degrees Celsius, coincides with the low temperature in the season. On the other hand, we see a higher biomass for September and October, which correlates with the higher temperature we had during those seasons. And in terms of site, we saw a high biomass output at WWD, which is where the effluent joins the river. As we move upstream and downstream from WWD, we saw a lower biomass in our samples. So based on our data, we attributed this difference to the carbon-nitrogen ratio, which tells us the amount of nutrient availability. Due to the wastewater effluent joining the river at WWD, we have a high nutrient availability, which fosters bacterial growth, thus yields a higher biomass in that specific site. However, if you might notice that in contrary to what I said, we have a high biomass record for OCB. After doing our data analysis, we were able to see that OCB is located nearby an agricultural land, which might expose that specific site again to a high nutrient availability and high biomass as well. But overall, looking at our microbial biomass, we were able to conclude that the effluents definitely impact the microbial biomass by providing higher nutrient availability, therefore increasing the growth of bacteria in the river. Maddie will be now sharing the other parts of our results. And meanwhile, I would like to encourage you to send your questions on our presentation. Thank you.
So this is our principal component analysis result, and this is looking specifically at the sedimentary bacterial community. It is a little bit more complicated of a figure, but essentially what we're seeing in this figure is that our winter samples that you can see in white are more similar to each other and have a more similar bacterial community than compared to our summer and fall samples. So we're seeing sort of a seasonal difference in terms of bacterial community. Could we go to the next slide, please? And then if we break up our principal component analysis even further, if we look on the left, we have all of our winter samples, and we're seeing 3 sort of groups of 3 groupings. In group 1, we have our nature park, which indicates that those bacterial communities in the nature park are more similar to each other than they are to any of our other sites. And then if we look at group 2, we're also seeing that our control site is more similar to those samples are more similar to each other in terms of bacterial community than any of our other sites. And this is good because this is our control, and it indicates that after our control point, there's something occurring in the river that's causing a change in bacterial community, and that would be the effluent point. And then if we look at group 3, we have our WWD, WWU, and our other downstream site grouped together, and they have similar bacterial communities, which indicates to us that since one of our downstream sites has a similar bacterial community to our effluent point site, that the wastewater treatment plant is introducing new bacteria into the river, and that's being sustained over a long period of time. If we look at the figure on the right, these are our summer and fall samples, and we're not seeing as clear of groupings as we did in the winter season. But if we look at our black diamonds and our gray diamonds, those are both our WWD sites for fall and summer, and those have a different bacterial community than the rest of the river, which still suggests that we're seeing an input of different bacteria at that site than anywhere else in the river. So the effluent still is introducing new bacteria that normally wouldn't be there. We do have 2 other groupings in this figure. One of them is the nature park in the summer, which is in the gray triangles. And then we also have a grouping of our control point and the nature park in the summer. These were a little bit odd data, but it is kind of just showing that we have some different bacterial communities, and there might be some other effects besides the wastewater treatment plant. But the major takeaway is that the wastewater treatment plant bacterial communities are way different than any of our other bacterial communities. If we could go to the next slide. And then this is our carbon utilization graph. And Amerti talked a little bit about how we got these results. There's 2 major takeaways from this graph. The first one is that despite seeing such large differences in bacterial communities, each site across all seasons are using the same amount of carbon source to similar percentages. So the bacteria are eating the same foods across site and season, which is interesting considering that we have such wide differences in bacterial community structure. And then another interesting thing to note is that for our nature park site in the winter, those bacteria were using carboxylic and acetic acids more, which is just an outlier in our data that we want to highlight. If we could go to the next slide. And then just some major conclusions from our paper is that the effluent is a major source of nutrients in the river. So the microbial biomass and bacterial composition varied seasonally regardless of effluent discharges into the creek. So on our PCA graph, we were seeing that in the winter, we were having different bacteria present than compared to the summer and the fall. And this can be due to temperature changes as well as changes in the carbon-nitrogen ratio that affect how many nutrients are available. We also see that our microbial biomass and bacterial community function vary depending on spatial positioning from the wastewater treatment plant outflow. So we saw that our control site in terms of biomass and community structure was a lot different than any of our other sites, which indicates that the effluent is affecting the river and causing a change in the amount of bacteria and the type of bacteria and other microbes we're seeing. And then we also saw further away from the outflow, the lower biomass was measured and we had higher carbon-nitrogen ratios. And this could indicate that because we're having such high biomass at the wastewater treatment plant effluent, that all of those microbes that are there are using a bunch of nutrients in the river, and those nutrients can't flow down to our further away sites, which could reduce their biomass, which isn't good because it's taking away nutrients from other organisms in the creek, which could have a really negative effect. And then we also saw lower metabolic diversity observed at the WWD site. So this indicates that less microbes and bacteria can survive in that area than any other site. So only specific organisms can live there, which also isn't great because you want high diversity because that's just better for the creek overall. If we could go to the next slide, please.
And now we'll talk a little bit about the publishing process, especially for us as undergraduate students. So starting with the experiment, after designing the project and making sure we have representative samples, we began our study. And then we employed the experimental procedures we discussed earlier. We analyzed our data and then used the appropriate statistics in order to represent it. Then after that, especially for me as a sophomore at the time, we did a very intensive literature review, reading a lot of papers, a lot of scientific papers, and then began writing the actual paper. We worked on this paper over the academic year too, but we put our whole summer into it, preparing the manuscript. We, with the help of Professor Akinwole, we were able to write each part of the paper and then go through it with him, get feedbacks, and then also learn the process of writing by self while experiencing the process. And we worked on it very hard. It looked amazing. We finished it. And especially as undergrad students, we were very excited. And then we submitted the paper. We waited for a month or so, and our paper was rejected. It was a very disappointing and devastating experience, but also a very humbling and inspiring experience as an aspiring scientist. We were able to work on it again, finalize it, and then submit it. And after multiple peer reviews, we were able to get it accepted. And a good lesson for me, and also I believe speak for Maddie and Sophia, was that there are challenges and also it's not easy, but the fact that we were able to do it taught us a lot in this process. It was frustrating for us, but I would really want to thank Professor Akinwole, who was really working so hard, being a role model for us, encouraging us and being calm through the process. As a person who even experienced that, and especially thinking that it was the best, it was a really good paper, and then getting it rejected wasn't motivating enough to start working on it again. So again, I really want to say thank you to Professor Akinwole for believing in us, working with us, and also helping us have a voice in this research and contribute to a greater impact in our community. I'll leave the floor to Maddie to share experience of this work as well.
Yeah, I think the biggest thing for me was I sort of knew the process of having to submit our manuscript and then waiting for peer feedback. But I just didn't realize how long it would take because it took over a year from when we started writing to when it finally was published. So it was a really, really long process. When we did get rejected, they send you back like a whole list of everything you basically did wrong and like corrections they had. So we pretty much had to like revamp a lot of the paper. We took away figures, we added figures, we made new figures. Yeah, so we had to add to like basically every section of the paper. And then also we were lucky enough to have grants and scholarships and funding that allowed us to publish, because publishing is not a free process. It's pretty expensive. So even for the paper that we didn't get submitted, we still had to pay for them to review it. And then if you do get accepted, you have to pay even more for it to be published. So we were thankful enough to have grants for that, which was really nice. And it was a great experience for me.
And along with some of the things that Maddie and Amerti mentioned, I would have to say that after you publish a paper, you gain a little more empathy for the authors that you read about in primary articles. And as a STEM major, you tend to read a lot of primary articles. So it does feel a little different having been published and understanding the process. Do you mind going to the next slide? And I also want to say that I have had such an incredible experience doing research at DePauw. Not only have I been able to work with an amazing group of professors in the biology department, but I've also been able to work with and learn from faculty and staff from other departments. It takes quite a diverse group of people with different backgrounds and different areas of expertise to properly work on and understand a project. Research experiences also allow for networking and collaborating with different departments, and starts to really build a community with these incredibly knowledgeable faculty and staff. I will always be thankful to them for helping to find funding, because I know professors aren't always on the largest grants, and I know there isn't a research fund in the biology department in particular. But I'm incredibly privileged to have had this research experience, and it was because of my time in Professor Akinwole's lab that I was able to apply for a national grant this past fall by using my scientific writing skills and to write about all of the different lab techniques that we used and what I would use in a hypothetical research project. And I can probably speak on both Amerti and Maddie's behalf that we all have a whole laundry list of techniques and lab skills that we know how to perform. And that looks amazing on a resume or CV. In research here at DePauw through semester credit research, through different classes and through paid summer research has been what has supported my continuation and interest in STEM and helped to shape and define my career path. Thank you. Could you go to the next slide, please?
And then these are just some people that we would like to thank for helping us do this project. DePauw students Anya Sanders, Natalie Michaels, and Emma Arndt for helping with sample collection and processing. Steve Taylor for the initial site survey. Wendy Tilmichael of the biology department for all of our chemical preparation. And then our funding was provided by the Asher Fund in the Sciences, FDC, Student Faculty, Collaborative Research, and the DePauw SRF Science Research Fellows Program.
Awesome. Well, thank you so much, you all, for presenting that research. It was fascinating, and it was really interesting to learn about that publishing process because, you know, not a lot of us have experience in that. So it's good to hear it from your perspective, and for anyone who might want to pursue that in the future, they now have that information, which is really helpful. So we can now open the floor to questions. So if people have questions that they want to submit, they can come in through the Q&A. You can ask them about the research, what it felt like to do the research, publishing, anything. Any and all questions are welcome. And while we wait for questions to come in, I have a question. So my question is, how do you think, other than a resume builder, how do you think the scientific processes and techniques that you've learned, how do you think they will help you in your future career paths, other than resume builders? And you all can answer.
Yeah, I can answer that question. So if you're a person who wants to go into research, which is what a lot of STEM students want to do, it's really nice to know the lab techniques before you start working a professional career. So it kind of gives you a little bit of an advantage and a boost. And also doing research has just made me more comfortable in the lab. So I'm like not as scared to like mess things up or that I'm like doing it wrong. So it really helped build my confidence and made me realize like I can do lab work and be successful in it. So that really helped encourage me to like continue on this career path.
Yes, I agree with Maddie as well. But also in future career, especially in terms of like designing our own experiment or addressing an issue, it gives us a great boost of energy because we've used some techniques, we know how to navigate them, or we know how to like tweak them to the process we want to apply. And just getting familiar with those processes make us more flexible and also very creative in designing our future projects in an efficient way as well, right? When we design an experiment, of course, we want it to be successful, but also we don't want to waste a lot of resource trying things. I'm not being against trying, but also just having the undergraduate experience of this will help us design like a more efficient and targeted research in the future.
Yes, and I agree with both Maddie and Amerti on that. You definitely gain a lot of confidence doing research. Like you have to be so okay with something not going the way that you want it to go. So having that like life lesson is definitely very valuable.
Well, thank you. Now, this can be for all four of you. Someone asks, do you think you would find the same results if you sampled other streams and rivers throughout Indiana or other states? Which is, I think, is a good question. So whoever wants to go first, you can go ahead.
Okay, I can start and anyone else can add on it. It's a very good question because we've already started working on it, and our next manuscript will be on a different river receiving effluents from various wastewater treatment plants. So we will be addressing this question in the manuscript and also looking at, it's not Indiana, it's in Illinois. It's a larger river. And the effluent is also released from a wastewater treatment plant that uses chlorination instead of UV. So I think Sophia has tried to touch upon it earlier. The Greencastle wastewater treatment plant uses UV radiation as a tertiary method of cleaning water. And the river that we're looking at now, recently in Des Plaines, Illinois, will be receiving effluents from a wastewater treatment plant using chlorination. So it'll be really nice to see if the impact will be the same, different, and all that. We're looking into it. The paper will be hopefully published soon, and maybe we'll meet you on a different presentation. But if anyone wants to add up on it, I'll be happy.
Okay, that's a good question. So generally, the environment we select for the type of bacteria that you have. So depending on the type of nutrient, the type of heavy metal, the type of effluent, now we influence the bacterial community, a different station. So we're not going to see the same. So if you sample different streams that have different parameters, then you will have different bacterial community. If they have the same parameter, then you will likely see the same bacterial community. So it depends on the environmental variables. So like Maddie said, right now we're looking at another river system that have a different type of effluent, and we don't expect to see the same type of bacterial community. Yeah. Thank you.
Awesome. Okay. Somebody else wants to know, what type of information on the Big Walnut Creek were you looking for before starting your research? Was it historical data? Was it, ecological data? And where did you look for it?
Okay, so initially we're trying to see any form of ecological data on the Big Walnut Creek. And I believe there's only one paper written several years ago, and they basically talk about the type of fish that reside in the Big Walnut Creek. So we don't have a lot of data on microbial community. On the maybe ecosystem functioning. So, and then as a microbial ecology, I'm kind of interested in how we impact the microbial community that perform a lot of the ecosystem function that are so important to us. So that was why we kind of started that work. And also because of the effluent that comes from the wastewater plant, we want to see how that also impacts the Big Walnut Creek ecosystem. And also we want to look at the possibility that we have pathogens or E. coli, and is it okay to have full contact with the Big Walnut Creek river? Can people swim safely in the water? So those are the things that kind of generate this study. Thank you.
Okay, somebody else wants to know, do you think your other DePauw classes, not STEM related, helped your writing processes, and how? So did your other DePauw classes help your writing? I feel like I know the answer, but I'd love to hear your opinion on it.
So scientific writing is a lot different than English writing or any other sort of writing you would do. It did help in terms of like grammar stuff, but I think a lot of English classes you kind of are sharing your own thoughts on papers and like your interpretation of a reading, whereas in science you kind of just have to give facts and statements. So in terms of like grammar and structure, it helped. In terms of helping with like writing the actual content of the paper, not so much. I think it was actually harder to transition from writing as I would in any other class to writing a strictly scientific paper.
Yeah, and I would have to agree with Maddie on that. And yeah, it's very, other classes are very good just for like, you know, going over drafts and whatnot of different papers that you might write and just having that practice of writing. But scientific writing is a whole different ball game. I actually, last semester, I took a course that was very heavily based on like scientific writing. It was a science research fellow course. And it just, taking a class like that really helps you with your scientific writing and helps you to convey yourself to either a more general population or a more scientific community. Being able to switch in between the two is really important as well. So.
Awesome. Okay, we have another science question. How were the upstream and downstream sites determined? And do you know the amount of flow from the wastewater treatment plant that was being discharged to the WWD sample point at the time of sample collection? I can repeat that one more time. So how were the upstream and downstream sites chosen? And do you know the amount of flow from the wastewater treatment plant that was being discharged to the WWD sample point at the time of collection?
So basically we try to look for a control site so that we can compare the other side to our outfall. So we sample about 5,000 meter transits. Upstream is about 1,000 meters away from the outfall so that we know that there's no influence on our control site. So that was the Dunbar Covered Bridge. And then we also sample a little bit just above the outfall, the WWD, and also then a couple of 1,000 meters downstream, so we can monitor the effects over some distance downstream of the effluent. So that's how we determine the our sample location, so that we can just kind of compare upstream and downstream to the actual point where the effluents kind of get into the Big Walnut Creek. So in terms of the flow, we only ask for the amount of the flow that comes into the plant. but we were not interested in the flow that kind of go out of the that goes from the effluent. We just want to know how much waste actually comes into the plant. So that was the only thing that we measure. We didn't measure the outflow into the because we don't see if that we influence our sampling because we are only dealing with just a single outflow. As mean we're comparing several discharges, like maybe several outflow into the Big Walnut Creek, then we can compare the flow. But since we only have one that goes into the Big Walnut Creek, so we didn't measure that. Thank you.
Another question, are these, effluents and bad things coming out of the wastewater treatment plants, are they responsible for mutations or, problems with organisms in the stream other than the microbial communities? But like, are other animals dying because of this? Is this a big problem or is it only a problem related to the microbial communities?
I can start with that question. We only worked on microbial community, but definitely the impact is not limited to the microbial community. Especially there are a lot of research out there working on fish, aquatic animals, and the impact especially with chlorinated water being released into those aquatic bodies. So as mentioned earlier, one of the ways to treat water is chlorination. And even if the water is dechlorinated before being released, whether it's the chemicals that it's used to dechlorinate it or even the chlorine itself, the increased amount of chlorine itself in the water would, a lot of studies have shown that would affect the aquatic animals such as fishes and all those in the water. So it's not definitely limited to bacteria. It just wasn't the scope of our research, but there are a lot of studies out there on it. And thank you.
In addition to that, so whenever we see changes in microbial community, it is an indication that there's also a problem at higher level. There could be accumulation of chemical in other organisms. So it can tell us maybe there are pathogens in the water that might influence aquatic life. Maybe there are metals that could be accumulated. And then if we consume fishes like that, then that can also be transferred to our body. So it's an indicator because the microbes are at the base of the food web. And so when you consume several of them, then they can accumulate in your body, or where they can accumulate in the fishes, then we can consume the fishes, they can accumulate in our body. So it's kind of like a spiral kind of process, yeah. Thank you.
Awesome. So we have time for one more question, and someone wanted to know, what are your paths after DePauw? What are you going to do after DePauw? So go ahead. Especially, I guess the seniors, obviously, that's up first.
So I currently, my career path is in the works. I'm really interested in working in national parks for maybe a couple of years and then find my way back to graduate school for restorative and conservation type of research, or do a program of that type.
To go to pathology school. I want to get a master's in pathology assistance. Specifically, I want to be a clinical pathologist. So I'd be the person performing autopsies. Police station.
Okay, so that was a little muffled, Maddie, but what I got was you wanted to go and get your master's. and pathology was the main interest. Is that correct? Yay, cool. Okay.
Oh, and for me, I'm currently a junior and looking into graduate schools, a PhD program in infectious disease, microbiology, and immunology are the areas I'm currently interested in.
Awesome. Well, thank you so much. Before we go, we just wanted to give some, last minute remarks. First, if you were viewing this event tonight, you will be sent a post-event survey tomorrow to let us know if you enjoyed learning about this student's research and what other sorts of events you would like to see in the future. And we invite you to stay involved with DePauw. We're grateful for your attendance tonight. It really is great for our students to see that alumni care about their research. And we invite you to continue to share your time, talents, and treasures. And you can do that by, getting involved, volunteering, giving up your treasures if you want. We know that, even the students said that a lot of this research couldn't be done without the help from our alumni. So that is super beneficial. And you can check out our website at any time to find out what sorts of opportunities are available for you as alumni. Speaking of events that you can be involved with, we do have some events that are coming up. On February 24th from 6 to 7 p.m. we have the final Alumni College of February, and that will be with Dr. Emmett Riley. And he will be talking about how the pandemic has magnified the disparities in healthcare. So that should be a really interesting talk. And then walking into March, on March 1st, we have the McDermott Center Speaker Series with Laura Chio, 98, and she is the U.S. Immunology Strategy and Operations Lead at Bristol Myers Squibb. And then on March 10th, we will be having an in-person event in Cincinnati for the Cincinnati region. And we're really excited about that as well. So as we continue, we invite you to stay involved and hope to see you on screen or on campus soon. So thank you all, and thank you to our wonderful students and researchers and our professor, obviously, for helping us with this presentation. Have a great night, everyone.